Hello guys, part 3 of our pharmaceutics 1 of your old syllabus D pharmacy. Our 11th question is write a note on Ayurvedic dosage form. Many times this question asked in your examination. So, what do you mean by Ayurvedic dosage form? See, Ayurvedic medicines, these are the medicines intended for the internal or external use for or in the diagnosis, treatment, mitigation or prevention of the disease or disorder in human beings or animals. Means these are the type of medicines. Why they are using? Either they are used for the internal use or the external use. Okay, or also they can use for the mitigation purpose, prevention purpose, diagnosis purpose. Okay, are you getting? Yeah. Now this Ayurvedic dosage form are obtained from the natural source, that is from animal, plants and minerals. These Ayurvedic drugs, they are obtained from the natural sources. Now, Ayurvedic dosage form, these are the different types of dosage forms classified into 4 groups. One is a solid dosage form, semi-solid, liquid and powder. In solid dosage form, pills, gutika, vatika. Examples: semi-solid, avleha, paka, lepa, then gharta. Then liquid dosage form, asava, arishta, arka, taila. Powder, bhasma, satwa, mandura, pisti, parpati. Just to remember churna, you remember 2 examples at least. Solid dosage form. Solid dosage form, here vatika or gutika. Medicaments in the form of tablets or pills are known as solid dosage form in Ayurveda. Example is vati or gutika. Here liquid dosage form, these are the medicine preparation prepared by the soaking the drugs in the powdered forms or in the form of their decoction in the solution of sugar or jaggery as have intended for a specific period of time. If may produce a self-generated alcohol on fermentation and have sedation. Liquid means they are liquid in nature. Decoction, do you observe the decoction? Decoction means see, just I will give a beautiful example here. You know the process of a tea making, how the tea get prepared, before adding milk to that tea, decoction is there. Why? That is black color tea, what we are calling. Means these are the liquid dosage forms. Black color like tea. Write a note on filtration and aids. What is this filtration and filter aids? Means filtration, it is a process of a separation of a solid from a fluid by passing the same through a porous medium that retains the solid but allows the liquid to pass through. It is a process. What it will do? In this process, the solid particles will retain or will remain on top of this filter aids. Then the liquid will collects in the container. Filter aids. These are the filter aids. The objective of filter aid is to prevent the medium from becoming blocked and to form an porous cake, hence reducing the resistance to flow of the filtrate. The main objective of filter aid is to prevent the blockage and also to prevent the cake formation. Filter aid forms a surface deposit which screens out the solids and also prevents the plugging of supporting filter media. Basically, these filter aids, they are on the surface and they help for to trap the solid things. And characteristics of the filter aids, some of the characteristics are there. See example, they are chemically inert and free from the impurities. Low specific gravity, so remain suspended in liquids. Porous rather than dense, so that previous cake can be formed, and they are recoverable. Disadvantages: remove the colored substances by absorbing them. Sometimes active principles such as alkaloids are absorbed on filter aids. Sometimes these type of the medicines, what they will do, they get absorbed on the filter aids. Rarely filters are source of contamination such as soluble iron salts, which can provoke degradation of the sensitive ingredients. Sometimes, but not all. Sometimes these filter aid may provoke to the contamination. Next here is an example for filter aids. See, datomite obtained from natural siliceous deposits, and perlite, it is an aluminum silicate, cellulose, asbestos, charcoal, talc, bentonite. These all are the filter aids. They play a vital role to remove the impurities as well. Next example is of here, next is of write a note on metafilters. Metafilter means it is a equipment used for the filtration. Metafilter functions as a strainer, means surface filtration for the separation of the particles. They will separate the particles. Then in this method, metal rings contain circular projections which are arranged as a necks to form channels on the edges. Means these metal rings are there, no? They plays a vital role here. They only trap all the impurities. Construction. See, this is the metal rings. This is all what you are observing now. These are the metal rings. And this is the construction of that metafilters. Outlet, pack adapter, collar, V-rod, metafilter rings, filter powder and the anti-cap. Construction. Metafilter consists of the series of metal rings. These are threaded so that a channel is formed on the edges, and it contains a grooved drainage column on which a series of metal rings are packed. These rings are usually made up of stainless steel, and each metal ring has a number of semicircular projections on one side of the surface. Okay, each metal ring has a semicircular projections. The projections are arranged the same way up. These rings are tightened on the drainage column with a nut. By using the nut we are tightening this one. The metafilters are also known as edge filters. If anyone asks, you write this one. Working, these filters placed in the vessel and may be operated by pumping the slurry under pressure or occasionally by application of the reduced pressure to the outlet side. It works how? By applying the pressure. Okay, just we are inserting that metafilters into the vessel by pumping the slurry under pressure. Then the slurry passes through the channels formed on the edges between the rings. This is slurry is there, no? That slurry passes through the channels formed on the edges between the rings. Slurry means what you want to filter, no? That slurry passes through these channels. Okay, channels, and the slurry passes through the channels formed on the edges between the rings. The clear liquid rises up and collected from the outlet into the reservoir. For the separation of the fine particles, a bed of suitable material such as kesal gur is first built up. The pack of rings serves essentially as a base on which a true filter media is supported. True filter media is also supported to prevent or to remove the varieties of the impurities or separations. Uses for clarification of the syrups, then filtration of injection solution, then clarification of insulin liquors. Advantages can be used. This can be used under high pressure without any danger of bursting the filter media, and running costs are low. It can be made up of a material resistant to corrosion to avoid contamination of the preparation. It is an extremely versatile filter, and sterile products can be handled. Next, write a note on percolation and maceration. See, percolation, the process in which the comminuted drug is extracted of its soluble constituents by the slow passage of suitable solvent through a column of drug. Here it is a process in which one drug, that is a comminuted drug, is extracted. It get extracted of its soluble constituents by the slow passage of a suitable solvent through a column of drug. Example, in coffee percolation, soluble compounds leaves the coffee. Okay, they will leaves the coffee and join the water. Insoluble, those are insoluble, no? That insoluble components remain within the coffee filter. You know that. Okay, tea cups, no, not tea cups, a tea bag. That tea bag, you observe in that tea bag, all this tea bag, see this tea bag, it contains the tea powder inside this. When you dip that tea powder where into the milk, means what happens? This small ingredients, powders, what they will just enters into this tea, means that milk, and make that milk as a tea, and the all solid particles remain here where it remains in this tea bag only. Just we are getting the essence of the tea and the taste of the tea. Okay, are you getting? This bag contains the insoluble components that remain within the filter, coffee filter, here tea filter example. The process of percolation includes 5 steps: combination of the drug, inhibition of drug, packing, maceration and percolation. Maceration process, various types of maceration process. The process for a tincture made from organized drugs, okay, this process is called simple maceration. Then a process for tincture made from unorganized drugs, okay, such as oleoresins and gum resins. This process is known as maceration with adjustment. Then preparation of water for injection. See, water is considered most important vehicle in the pharmaceutical industries. Water. And pharmaceutical water is the most important of all the pharmaceutical utility. It is used as excipients in many pharmaceutical formulation as a cleaning agent. Now here water for injection, it's prepared from distillation or any other process, but distillation, it is a common process for preparing the water for injection. Superior than distillation for removal of chemicals and microorganisms, but is also pyrogen free. Which one? The water for injection is there, no, that is pyrogen free. Intended to be used for the parenteral preparation, means for all parenteral, for all IV routes, muscular route, we are using this type of water only. Then intended to be used for 24 hours after its collection, must be protected from the microbial contamination. Construction on steam distillation. See, one of the very important one, steam distillation. This steam distillation is also used for the preparation of the water for injection. So distillation is the process of separating the components or substances from liquid mixture by using selective boiling and condensation. Just what we are doing means we are separating the liquid mixture. One liquid contains 2 or more components. We are separating that with the help of the boiling and the condensation. And steam distillation, a special type of distillation. Temperature sensitive materials like natural aromatic compounds can be distilled here. Once it once was a popular laboratory method. Purification of organic compounds. Distillation at lower temperature reduces the deterioration of the desired products. Then principle of this steam distillation. When a mixture of two practically immiscible liquids is heated, liquid exerts its own vapor pressure as a function of temperature, as if the other constituents were not present. Simple, if 2 or more immiscible liquids are present means they are heated, and if 2 or more liquid we are using and we are heating the 2 or more liquids, in that what happens, one liquid is there, no, that one liquid, it gets suddenly heated and it get vaporizes, means one is vaporized, one is vaporized and collected in another container, one more is remains, means we separated, no, yes, simple, with the help of the boiling point. And vapor pressure of the whole system increases. Boiling begins when the sum of the vapor pressure of the two immiscible liquids just exceeds the atmospheric pressure. Boiling begins when it exceeds the atmospheric pressure. For an example, you take, what is the boiling point of water? 100. If it exceeds the 100 means then only boiling takes place. In this way, many organic compounds insoluble in water can be purified at the temperature well below the point at which decomposition occurs. For example, the boiling point of the bromobenzene is 156 degree and the boiling point of the water is 100 degree. See, if you heat these two components means on 100 degree Celsius, what happens? Water starts evaporating. But here 156 degree bromobenzene, it won't start getting evaporated. So we can easily separate this one. But here both the mixture of these two boils at 95 degree Celsius. I told you water boil at 100 degree and that bromobenzene at 156. But when we use both these, they get boils at 95 degree Celsius. So that we can easily separate. Thus bromobenzene can be easily distilled at the temperature 61 degree below its normal boiling point. This one is apparatus. You just check out this apparatus. Contains the condensed water vapor, then water outlet is there. Distilled liquid water is there. This one is the water and all. Check it. Construction on round bottom flask, a glass inlet tube which reaches almost to the bottom of the flask, steam generator, water condenser, then additional steel head, then receiver. Application: it is employed in the manufacturing of essential oils, perfumes, eucalyptus oil, orange oil. It is used to separate the intermediate or final products during synthesis of complex organic compounds. Also widely used in the petroleum refineries and the petrochemical plants. Study of tray dryers is a tray dryer is a device used for the drying of the wet products of the crude drugs, chemicals, powders, or the granules. This one is a construction of the tray dryer. Okay, construction of the tray dryer. It consists of the rectangular chamber of the sheets metal containing two trucks that support rack hatch. This is the rack hatch. Okay, two rectangular sheets are there. Each rack carries a number of shallow trays, perhaps 750 millimeter square, 50 to 150 millimeter deep, that are loaded with the material to be dried. What we want to dry, we are keeping on these trays. Heated air is circulated at 2 to 5 meter per second between the trays by fan C and motor D and passes over the heaters E. With the help of this fan C and with the help of the motor D, okay, with the help of the fan C and motor D, what happens? The heated air, what it will do, it is circulated here in the trays. Okay, you are observing these arrow marks. This is the circulation of the heat. Buffels G distribute the air uniformly over the tray of, this is the buffels, they will distribute the air uniformly throughout the trays. Some moist air is continuously vented through exhaust duct B, make up fresh air enters through the inlet A. Okay, this exhaust B is there now. Go to this exhaust B, it will remove the moist air and it allows the new air, okay, new fresh air through the A inlet. Then the racks are mounted on a track wheel I. See, this is the track wheel I. The racks are mounted so that the end of the drying cycle, the truck can be pulled out of the chamber and taken to the tray dumping station. Okay, easily we can remove that all. Working of the tray dryer: wet solid is loaded in the trays and trays are placed in the chamber. Fresh air is introduced to the inlet which passes through the heaters and gets heated up. The hot air is circulated by means of fans and then water is picked up by the air. Then these events occur in the single pass of air. The time of contact is short and amount of water picked up in a single pass is small. Then at the end of the drying trays, trucks are pulled out of the chamber and taken to a tray dumping station. Advantages: tray dryers are very useful when the production rate is small. They find most frequent application for vulnerable products like dyes and pharmaceuticals. In the tray dryers, the handling of the materials, loading and unloading of the materials can be done without losses and energy saving. Okay, very important. Here energy is saved. Fluidized bed dryer. Okay, fluidized bed dryer. Fluid bed drying is the most widely used technique for drying pharmaceutical powders and granulation. The direct contact between the particles, air and gas, is possible in fluid bed system. Here any type of the inert gas is used. Why we use an inert gas? See, inert gas is used here in a fluid bed dryer, fluidized bed dryer or fluid bed dryer. Okay, very important thing. See, this one is one construction for a fluid bed dryer. Working: when the velocity of air is greater than the settling velocity of the granules, the granule remain partially suspended in the gas stream. See, same it works on the principle of the cyclone. Okay, how the air get trapped means how the particles are hit on the wall of that cyclone separator. No, here also velocity of the air is greater. What we are adding, that air should be greater than the velocity of the granules. Then the granules will partially suspended in the gas stream. After some time, a point of pressure is reached at which frictional drag in the particles is equal to the force of the gravity. The granules rise in the container because of the high velocity gas and fell back in the random boiling motion. This condition is said to be fluidized state. Drying is achieved at constant rate and falling rate period is very short. Very simple. See, those having greater velocity, what they will do? Okay, they will float at the top. Those having lesser velocity, they just float at the downside, bottom side. Advantages: fluidized bed dryers. They requires these are type of dryers which helps to dry the equipments. They dries the less, means in a very less time the drying can be completed, 20 to 40 minutes. Hot spots are not absorbed in the dryer because of its excellent mixing and drying capacities. The thermal efficiency is 2 to 6 times greater than the tray dryers. It facilitates the drying of thermolabile substances. Since the contact time for drying is short, it can be used either batch or the continuous type. Moist heat sterilization. See, principle: denaturation and coagulation of the proteins due to the latent heat present in the steam. With the help of the steam, with the help of the heat in the steam, we are heating the products so that the products can be free from contamination. Autoclave: it is a type of moist heat sterilization. Autoclave, it is a pressurized device designed to heat aqueous solution above their boiling point at normal atmospheric pressure to achieve the sterilization. Principle: boiling point of water, it is directly proportional to the pressure. Okay, greater the boiling point, pressure is more when the volume is constant. Pressure is directly proportional to temperature. When pressure is increased in a closed vessel, the temperature increases proportionally. That is then next point, this pressure and temperature is kept constant for 20 minutes during autoclave. It is sufficient to kill the vegetative forms and spores of the organisms. For 20 minutes, if we keep the temperature constant, means we can easily kill. Construction: see, this one is the exam diagram for this autoclave. Go through this diagram. Autoclave diagram. Construction: here a cylinder vessel made up of gunmetal, controller and time, and with time and pressure programmable by user. A backlit alphanumeric 2-line 32 character LCD display. LCD display is also there. Low water level sensor is there. Then lid is filled with the pressure gauge, safety valve. These all pressure gauge, safety valve, safety plug. Okay, manual exhaust valve, vacuum breaker. Then lid ensure an airtight closure in the autoclave. A perforated plate which is used for keeping the material to be sterilized. Drain valve for easy drainage and cleaning. Molded rubber gasket and stainless steel carrier along with the heater cover stand. Autoclave microbiology we are using, medicines we are using, veterinary science, dentistry and metallurgy we are using this autoclave. Okay, then dry heat sterilization. Example here is a dry heat, that is hot air oven, and oven provides a temperature higher than that of atmosphere. The temperature range covered by ovens is between 50 to 250 degrees Celsius here. Then these are used for the rapid evaporation of the materials and rapid drying and for sterilization of the articles that can be sterilized by the dry heat. Dry sterilization technique requires longer exposure time, that it requires 1.5 to 3 hours, and higher temperature than moist heat sterilization. Means greater temperature requires than the moist heat sterilization. Principle: sterilization by dry heat is accomplished by conduction. Then the heat is absorbed by the outside surface of the item, then passes towards the center of the item layer by layer. Then the entire item will eventually reach the temperature and required for the sterilization to take place. Dry heat does most of the damage by oxidizing molecules. The essential cell constituents are destroyed. Microbes, proteins of that microbes get destroyed. Okay, are you getting know what I am telling? Yes, the temperature is maintained for almost an hour to kill the most, means they are maintained, the temperature is maintained for 1 hour to kill most difficult bacteria or resistant spores. Then most common temperature here is for 30 minutes 170 degrees Celsius, for next 60 minutes 160 degrees Celsius, for next 150 minutes 150 degrees Celsius. Okay, and this is used for the glassware, forceps, swabs, water immiscible oils, waxes and powders to sterilize this all. This is the construction of the your hot air oven. Go through this one. Advantages: a dry heat cabinet is easy to install and has relatively low operating cost. It penetrates material, it is non-toxic and do not harm the environment. It is non-corrosive for metal and sharp instruments.
Define, classify, prepare compressed tablets. See, tablets are compressed solid unit dosage form containing medicaments or medicaments usually circular in shape, may be flat or biconcave. Advantages: they are easy to administer, easy to dispense, more stable, accuracy in drug, bitter and nauseous substances can easily be dispensed. Okay, disadvantages: problem with compression to crystalline drug. Crystalline drug, they cannot be compressed. Hygroscopic drugs are not suitable for compressed tablet. Hygroscopic, those drugs which provoke for the water, they are not used here because they form the stickiness. Then drugs with low or poor water solubility, slow dissolution, may be difficult to formulate. This type of the tablet, compressed tablets. Types of tablets: tablets injected orally, then tablets used in the oral cavity, tablets administered by other route. Oral tablet like complex tablet, multiple complex, delayed release, sugar, film coated, chewable tablets. Here used in oral cavity, buccal tablets, sublingual, lozenges and dental cone. Then tablets administered by other route like vaginal, implantation, is all. Manufacturing of the compressed tablets, preparation of granules for compression. Here wet granulation is one of the preferred method. Okay, is widely employed method for the production of the compressed tablets. Here the steps are weighing and blending the ingredients, preparing a damped powder or damp mass, screening the damp powder or damp mass into pellets or granules, drying that granules, sizing the granules by dry screening, then adding lubricant and blending, forming tablets by compression. Methods of tablet coating: different types of coatings are there, and main reasons for coating is to mask unpleasant taste and odor, to improve the appearance of tablets, to prevent the medicaments from atmospheric effects, to control the site of action of drug, to produce the sustained release product. Then different types of coating like sugar coating, film coating and enteric coating. Sugar coating, in this sugar coating, sieving is there, sealing is there, sub coating, syrup coating and finishing. Just you write these sub points, it's enough. Okay, sugar coating involves sieving, sealing, sub coating, syrup coating and finishing. Enteric coating, these tablets are coated with the material which does not disintegrate in the stomach but passes through as it is, that is enteric polymer. Hydroxy propyl methyl cellulose phthalate, these tablets dissolve in the intestine. Okay, it means those tablets which they want to directly show their effect only in the intestine, not in the stomach. So to prevent getting dissolutes or dissolved into the stomach, we are coating this one, that is enteric coated. Film coating means in these tablets are coated by the single or the mixture of film forming polymers such as hydroxypropyl methyl cellulose, hydroxyethyl methyl cellulose, methyl cellulose and all. And this the polymer is dissolved in some volatile organic solvent and is sprayed over the tablets in rotating pan. Note on capsule: capsules are the solid dosage form in which the drug substances are enclosed either in a hard or soft soluble shell, usually formed from gelatin. The term capsule is drawn from the Latin word capsula, meaning container. Types of capsule: one is hard and another one is soft gelatin capsule. Hard gelatin capsule, these are the capsules consist of the two pieces in the form of cylinder closed at one another. And the shorter pieces is called the cap. Shorter pieces, it is called the cap. On the top is there, no, that is shorter piece. And the longer piece, that is called the body down. And hard gelatin capsule, also called a dry filled capsule. The drug substances placed in the body and the caps are slided over it, hence enclosing the drug substances. Simple. The main important drug is narrow, that is stored into the body. Okay, for that body it is closed with the cap. Soft gelatin capsules are one piece hermetically sealed soft gelatin shells containing liquid and a suspension or semi-solid. Soft gelatin is mainly composed of the gelatin, plasticizers, preservative, colorings, flavoring agent, sugars and all. Write a note on vaccine. Vaccine is a biological preparation that improves immunity to a particular disease. A vaccine typically contains an agent that resembles the disease causing organism, microorganism, often made from the weakened or killed forms of the microbe. You know that vaccines, they are either killed or the inactivated form of the microorganisms. Just we are inserting in the human body or animal just to boost the immunity. Okay, types here: live, attenuated vaccine, inactivated subunit toxoid conjugate DNA recombinant vector vaccine. Live attenuated means, see here, the live attenuated vaccines contain the version of the living microbe that has been weakened in the lab, so it can't cause disease. It is living but weak, because a live attenuated vaccine is the closest thing to the natural infection. These vaccines are good teachers to the immune system. Okay, they helps to increase the immunity of the consumer or the patient. Vaccines against measles, mumps and chicken pox, okay, we are giving, no, before only we are giving these vaccines before chicken pox, because next time when chicken pox attack to us, no, at that time our body get resist to that chicken pox. Okay, it having good immunity. Yes, these are the important questions of pharmaceutics first year D pharmacy old syllabus. Go through these important questions with notes. Thank you.